Yeah, today we'll discuss about ketone bodies. It's an overview of ketone body synthesis, utilization, and the functions of ketone bodies, significance of ketone bodies. And this concept comes under lipid metabolism. It's a very important question for essay, 10 marks. So please try to understand the concept of ketone bodies. So what are ketone bodies and significance of ketone bodies we will see. So mostly these ketone bodies are produced in our body mainly in two conditions. Condition 1 is starvation, condition 2 is diabetes. In this video we will see the production of ketone bodies in starvation phase. So what happens in the starvation? When we are in the starvation, what does it mean? Starvation means there is no food, we don't take any food. So if we eat food, what is the chief source of energy in our body? Glucose. So glucose is a chief source of energy. Our energy is in the form of ATP. So if we don't take glucose, we cannot get ATP. Then what happens in our body? We need continuous supply of energy. For that energy we need a continuous supply of glucose or any other food, but we are not taking anything now. There is no glucose levels are very low. This low arrow mark indicates low glucose values and low energy levels. What happens at the time? So in our body there is a mechanism, there is a hormonal mechanism called, here we can see in the pancreas, if there is no food in our blood or no glucose in our blood, low levels of ATP, what happens in the pancreas? So in the pancreas is divided into two parts. One is a exocrine part and another is a endocrine part. And the tail part of the pancreas has endocrine part. So this endocrine part releases two hormones. One is insulin, and next one, other one is glucagon. So this insulin doesn't play any role in the synthesis of ketone bodies, but this glucagon plays a role in the synthesis of ketone bodies. So this glucagon releases whenever there is no glucose levels in the body, there is no energy levels, low levels of energy, results the release of glucagon from the endocrine part of the pancreas. So this released glucagon goes and acts on the adipose tissue. So what exactly happens once this glucagon acts on the adipose tissue? In the adipose tissue, fats are stored in the form of triacylglycerols, in the short form TAG. So fats in the adipose tissue are stored in the form of TAG or triacylglycerols. As soon as glucagon acts on the adipose tissue, these triacylglycerols are released in the form of diacylglycerols and monoacylglycerols into the bloodstream. So this is a blood vessel, imagine. So in the bloodstream, diacylglycerol, monoacylglycerol is there. Since these fatty acids are insoluble in a blood circulation, these fatty acids utilize a protein called albumin as a transporter. See here, so this is albumin which covers the entire structures of diacylglycerol and monoacylglycerol, and this albumin takes this diacylglycerol and monoacylglycerol to the respective area where exactly we get energy from these fats. So again I'm gonna repeat this whole thing. So whenever we are in the starvation or hunger strike, whatever you can say, fasting, starvation, whatever you can say, our blood glucose levels are will be very low, and the ATPs also will be very low. At the time, what happens? The pancreas releases glucagon hormone and it will act on the adipose tissue. Adipose tissue releases the fats in the form of diacylglycerol and monoacylglycerol. So these two fatty acids need to go liver in order to give energy from the fats. So in order to go to the liver, these should be transported via blood circulation, but since these are insoluble in blood, these fatty acids utilize albumin, protein carrier, from adipose tissue to the liver. So these fatty acids finally reach to the liver. So in the liver hepatocytes, a pathway called beta oxidation takes place. So this beta oxidation is a pathway which oxidizes these fats, diacylglycerols and monoacylglycerols, into acetyl-CoAs. Acetyl-CoA. So this produced acetyl-CoAs will be participated in the pathway called TCA cycle, and also these acetyl-CoAs are the sources for ketogenesis. So the ketogenesis means synthesis of ketone bodies. Keto means ketone bodies and genesis means synthesis. So these acetyl-CoAs are the sources, are the precursors for the ketone body synthesis. So at the end of this beta oxidation, acetyl-CoAs are formed. These acetyl-CoAs are the sources of ketone body synthesis. So at the end of this ketone body synthesis, we will get three types of ketone bodies. Number one is beta hydroxybutyrate, and number two ketone body is acetoacetate. Number three is acetone. See here why all these are ketone bodies, because the structures of acetoacetate and the structure of acetone consists C double bond O group. Since this group is keto group and the presence of this group in these two ketone bodies, hence these are ketone bodies. But this beta hydroxybutyrate is also comes under ketone bodies, but this structure of beta hydroxybutyrate lacks the ketone body, keto group. So that's why these two are true ketone bodies, whereas beta hydroxybutyrate is not a true ketone body but ketone body. So we'll explain the significance of all three ketone bodies. So as soon as these ketone bodies are produced in the starvation by the action of glucagon, these ketone bodies, so this acetone ketone body will go to the lungs, and this acetone will be exhaled via lungs. This ketone body acetone exhaled via lungs, hence it is volatile. So since the presence of acetone in the breath gives the breath smell kusumol breath. So in the breath this acetone will be present. So since this acetone is present in the breath, if you smell the breath of acetone containing breath, you will feel kusumol breath, or also you can say fruity odor breath. Generally we observe this fruity odor or kusumol breath in diabetic patients, which we discuss in the next video. Diabetic patients. So move on to the next ketone body, acetoacetate. So this acetoacetate will be excreted via kidneys. So excreted by the kidneys, acetoacetate. So this acetoacetate will be seen in urine. You can find out this acetoacetate by performing Rodera's test. So Rodera's test gives you a positive result. If acetoacetate is present in this test, it will give you a ring in the test tube, indicates the presence of acetoacetate ketone body. And what about this beta hydroxybutyrate? The ultimate aim of ketone body synthesis and beta oxidation during starvation is getting energy from the fats. So this hydroxybutyrate remained ketone body will undergo ketolysis. Ketolysis means degradation of this beta hydroxybutyrate. So as soon as ketolysis occurred, ketolysis will give us acetyl-CoA again. So this acetyl-CoA, once this acetyl-CoA is formed, this acetyl-CoA will involve in TCA cycle, undergo TCA cycle. Once the Krebs cycle is done, we'll get energy in the form of ATP. So this is what the ketone body synthesis is in the starvation. Thank you for watching.